Hello everyone, welcome to MES e-learning channel. So in this lecture, we will be talking about what is genome editing. This would be a series of lecture. In the first one, we will talk about the basic mechanism for genome editing. So let us first understand what is the need and of genome editing. So as we all know that the invention of DNA sequences and the mechanisms in which we can precisely manipulate these DNA sequences have been very useful for various reasons like in agriculture, in medicine and in gene therapy. So manipulating these DNA sequences precisely is very, very important. So these processes will involve some specific enzymes which are called as your nucleases, and these nucleases should be able to target some specific sequences. So they will break the DNA, cause your double-stranded breaks, and then we may insert some sequences or we may delete some sequences, delete for causing your knockout animals which could be useful for studying a lot of different genes and other purposes. So understanding this mechanism is very, very crucial for the researchers and the professionals in the field of molecular biology, for genetics as well as gene therapy. So for understanding genome editing, it is very much important that we understand what is your double-stranded breaks and how the repair are being created, or how we can repair such kind of your double-stranded breaks. So there are two mechanisms for this repair mechanism. One is your homology directed repair mechanism. So when we are talking about homology directed repair mechanism, so if you have your double-stranded DNA and this double-stranded DNA gets broken up, so you have this kind of your double-stranded break that is created. So if we are adding a target molecule or a target DNA sequence, so this target sequence can actually get repaired, or this particular DNA which is there, the original DNA will be repaired incorporating this target sequence that is there. So for homology directed repair, it is very much important that we keep this target sequence, okay. So this all things would be very much important in case of gene therapy. So gene therapy where our original sequence is actually having some kind of mutations. So in that case, we want that that mutation has to be substituted by a normal working gene. So in this case, this target gene would be your normal working gene that would be actually substituting the mutated portion of your original gene in the sample. So this is your homology directed repair mechanism where we will be requiring this target sequence. Now there is other type of your mechanisms also, like where you have your non-homology and directed repair, so that is called as your NHEJ. So it is just non-homologously end joining. It is just joining the bases. So when you are having this double-stranded break, in this cases, what happens is arbitrarily there could be some overhang regions. So these overhang regions could be deleted, okay, during the repair mechanism. These overhang regions are deleted, and using your ligase, what is going to be done is these ligases are going to just occupy, these ligases are going to just connect the ends of this particular DNA together to repair this DNA sequence. So this is called as your end joining. So such kind of your NHEJ can actually cause some kind of your either deletions or insertions in this case. Okay, so these kind of your deletions and insertions could be very much useful if we want to develop some kind of your knockout animals, okay, or your knockout genes, okay. So in this case, what can happen is that the original sequence which is there, the original sequence is actually a functional gene, and then that functional gene we are trying to disrupt it, okay, disrupt it by introducing some kind of your deletions or insertions, okay. So disrupt that gene and cause a knockout gene. So these are the two mechanisms which are very important and would be common in all the mechanisms of your DNA editing. So whenever you are doing your genome editing, there would be one nuclease which would be cutting it, and then so after the double-stranded break has been caused, okay, by those kind of your nucleases, then it would be followed by either your HDR or it would be followed by your NHEJ. Either of this would be followed. So depending upon what is the purpose of your genome editing. So moving ahead, as I have already told you, that we can have either your gene knockout or mutations using different techniques like NHEJ. So where we can introduce such kind of your small insertions or deletions, or we can have your gene deletions also. So here you can have your two double-stranded targeted double-stranded flanking of your genes, and it is able, like with the help of your those HDR mechanism, we can delete a large portion of the DNA also. Okay, so that is also possible, or we can go for the gene correction. So gene correction, as I said, that already if there is some kind of your mutation in your target gene, then that could be replaced by your original working gene. So applications of genome editing is huge. So first is disease treatment. So that is in case of your gene therapy. So whenever there is any kind of your genetic disorders, okay, having mutations, those can be replaced by the corrected genome copy. Then we can go for the crop improvement. So in agriculture, genome editing plays a very important role. Okay, so it can enhance the different traits which are there, like yield improvement could be there, it could be disease resistance, or any kind of traits that help in enhancing the nutritional value of that particular crop, that can be incorporated in agriculture. Then, as we have already talked about, animal models, which are very important for developing different types of treatments. Okay, so for drug testing, animal models play a very important role. So for that, creating of the animal models also, a lot of genome editing is going to be required. And then in basic research, so where we want to understand the function of a particular gene. So these functions could be easily understood with the help of these kind of your mechanisms. For example, let me tell you, like if all the genes are working normally in a particular cell, then it becomes very difficult to understand that which gene is responsible for which function. But if you are able to reduce the activity, or you are able to just knock out a particular or knock down a particular gene so that it does not give its function, then we would be able to understand that whatever the abnormalities that are being seen in a particular cell, that is corresponding to that functional gene. Because that gene was not functional, that whatever the disturbances, whatever the problems that are occurring, that is because of that. So moving ahead, let us understand that what is the future of genetic editing. So the first one is technological advancement. Yes, as we are progressing ahead, new mechanisms of genome editing is coming up. There is development of this. What is the need for technological advancement? The problem with genome editing is the precision. If we are not having precise targeted genome editing, it will create a lot of problems. So we are looking ahead for new technologies so that we can have this precision, more and more accuracy we are able to achieve. Then for expanded applications, like there are tremendous applications. Medicine, we have already seen a lot of applications. Agriculture, and nowadays for environment and for conservation, and lot of things we are looking for. For example, in environment also having the biosensors. Okay, so that's also a very important application for genome editing. But all those things are fine, but we also have to look for the ethical considerations. As we all know that genome editing or changing the genomes is considered to be unethical in very practices, but because it is raising a lot of different questions, like whether the technology can be utilized in human beings and mainly in the germ lens. So if there is any kind of mutation that we are introducing in one generation, and if that is going to move on to the next generation, is it going to be safe? So all these questions have to be answered, and for that long term understanding of the effects is very much needed. So for that we require a lot of regulatory frameworks. Okay, so the government and internationally also there are a lot of different agencies which make policies on how we can, or on which cells or which models we can apply these gene editing models. And there are a lot of debate related to whether we should use it in the day-to-day life or not. So ethical considerations, as we were talking about the potential risk, okay, so long-term understanding of this technology is very much important, and for that ethical frameworks have to be designed so that for the benefit of the society, we should ensure that the genome editing is used responsibly. Okay, so for example, like we do not want any kind of your bio warfares, right? Because such kind of genome editing can also open the door for these kind of your mutations, which could be tremendously harmful for the human beings. So those kind of ethical frameworks are very much important, and having a public dialogue, that means we should openly discuss all those things. What is the implications of such kind of your gene editing techniques in the public, and discuss with the general public, so that they should be able to accept the changes, because wherever it is beneficial for the human beings, and accordingly the policies and regulations have to be in place. So this was all um, about the basics of genome editing. Further, we will be understanding about how these mechanisms, or what are the different mechanisms which are available that we can use it for genome editing. I hope I have cleared this small concept. Thank you.